This is question 5, component 3, lower 6 in class question book. All terrestrial plants, that means plants that live on the land as opposed to in the sea or on the seashore. They lose water from their leaves by a process called transpiration. That's fine. Water leaving leaves through stomata, you know that. A student used a potometer, so like you used a potometer, to estimate the transpiration rate of a leafy shoot over a 24-hour period. So you'll have done it over about 5 minutes possibly, but they did it over 24 hours. Explain why the water uptake from the potometer gives an estimate of the transpiration rate, not its true value. So water's coming out the top of the plant. That's what's powering the process, but we're actually measuring the water that's going in at the bottom, that's sucked up from the stem. What could possibly happen to water in between? Well, this is a plant, and plants photosynthesize, and we know the equation for photosynthesis is using up water. So photosynthesis uses water. So photosynthesis, remember H2O plus CO2 gives oxygen plus, you could balance this with 6's in front of these. So water could get used. And indeed in respiration, if respiration is the dominant process, water could be produced. That could be another reason. So this just measures the rate of absorption at the bottom of the plant rather than the rate of transpiration at the top. So one of those reasons. Nice 3 marks here. 3 other variables that have needed to be controlled when using the potometer. Two different plant species. You just need to be careful with what they're giving you already, and at the same light intensity and temperature. So I'd have loved to be able to say light intensity and temperature there, or indeed say keep the plants the same, but I can't use those. It has to be something else. So what else do I know? Well, I know my factors affecting transpiration. What can I? Yeah, humidity. That's one. Humidity. So I need to keep the humidity the same. And I remember using the hair dryers or wafting the plants in the lab. So that's the wind speed or the air currents. And then I'm stuck for a third then, because I can't say temperature, I can't say light intensity, I can't say keep the plant species same. But if I'm using 2 different plant species, I'd need to keep this fair, wouldn't I? So I'd need to keep the surface area of the leaves the same. So surface area of leaf the same. And then that would be a fair test. You might have in your practical, you might have worked out the surface area of your leaves either from approximating the geraniums as circles, or you might have cut out bits of graph paper and worked out per area. The table shows the average density. That means how many there are per unit area. So how many there are per square millimetre, and distribution, which means where are they on the leaves of oak and wheat. Looking at the table, the wheat has 50 per square millimetre on the one side of the leaf, the upper leaf, and 40 on the lower. So pretty similar. So slightly more on the upper. The oak, none on the upper, while lots on the lower. So wheat about the same or slightly more on the upper, oak all on the lower, none on the upper. That's the trend in the table. We need to explain this though. We need to explain. So this is really, we're thinking because. There's 2 marks here and there's 2 lines. If you start your answer, the distribution of the stomata in the wheat and the yolk is different because you've used a whole line up. So we don't even really need because, we're only writing because then it sort of gives you a clue as to what explain means. So let's talk about the wheat first. Well, it gives you a picture here. The wheat is, you know, almost vertical, you know, most of the leaves there, and so it doesn't really matter which side is the upper and the lower really. So that might explain why there's equal distribution. Whereas our oak here, it's definitely got an upper which has got the sunshine coming on, and the lower which is shaded. So that's our explanation. And so in wheat, neither side is shaded, so roughly equal distribution. And oak, lower surface is shaded, so will reduce water loss. Just trying to keep that in the space available. So this, if the stomata are on the top of the oak leaf, then presumably the water loss will be too much. Over the page we have got a new version of our stomatal diagram, so one that you might not have seen in the textbooks. The diagrams show guard cells in a leaf of wheat under 2 different environmental conditions. So where is the stoma here? It labels it, but it's not really. If I put yellow there, that's the hole in the leaf, that's the stoma. Whereas in the left diagram it's closed. What are the differences can we see? Well, this guard cell here, well, it appears to be there's more. It appears to be a higher volume of cytoplasm, more cytoplasm in this side. So if I colour it in this green there, those are bigger than the ones on the left. So it appears to be more cytoplasm. And then everything else looks pretty similar, and it'll be the same at the bottom. And that goes through there. So you can see what's inside the cell. There we are. Okay. Stomata are open when potassium ions are actively transported into the guard cells. Explain how this would cause wheat stomata to open. Right, well, 5 marks. How many lines have we got? 1, 2, 3, 4, 5, 6, 7 lines. So it's going to be tough to get all these 5 marks. And it's told us that potassium ions are actively transported, so we can't get a mark from repeating that. So we need to say immediately start off with what they do. So potassium ions, when they go in, they will reduce the water potential in the cell, because they're dissolved. So K plus ions reduce water potential in the guard cell. And I've spelled out water potential. I haven't put any symbols or WP or anything like that. I've said water potential. You could say other phrases, osmotic potential, solute potential, hypertonic, all this. Why don't you just say water potential like we've told you and shown you on the scale? Just say water potential instead of concentrations of things and all sorts. Now let's describe what happens to water. Well, water goes down a water potential gradient from high water potential to low water potential. And we just said we've reduced the water potential. So water moves in by osmosis down the water potential gradient. So water moves in by osmosis down the water potential gradient. And how many times have I heard that in exams? Millions of times. And that's the sort of thing we'd expect every time we're seeing a question like this. So water moves in by osmosis. We haven't actually said why the guard cells are changing shape or what's happening to open the stomata. Well, we've got our cells now becoming turgid. The pressure is increasing. So we've got a higher volume of cytoplasm because of the water, and this is increasing. So cells become turgid. Or you could say that the pressure increases. I'll put that in brackets. Pressure increases. We still haven't answered the question yet, which is how do these things actually open? Well, for help with this we need to look at the diagram, and we can look at the end of these guard cells. Well, the cell wall is thinner at the ends and is thicker in the middle. So just looking at the diagram, and we're going to actually get a mark for noticing that, but you should remember it from your notes anyway. So the ends of the guard cell have thinner walls than at the centre. Have thinner walls than at the centre. Or you could have said that the other way around. The centre has thicker walls than the ends. If I just show you that what we're talking about this area here. That's thick and thin at the end there. That's what we're talking about. And so, and this is the really difficult mark at the end. The ends of the guard cell expand and the stomata opens. Stoma opens. So when this gets bigger on this diagram, there's no other way of doing it. If you make the ends bigger, then these are gonna move apart. So the ends of the guard cell expand. Just really using this information. If you don't get this mark, your teacher will probably put UI next to it. Use this information in the diagram. And the stomata opens and stoma opens. So they've used stomata here, the plural, stoma singular, and really in exams you can use them interchangeably. Part D. Water is lost through stomata by transpiration. Okay, I think we're happy there. This results in a shell of water vapour forming over the stomatal pores. So this is upside down diagram where we've got the leaf. Where's my green? So we've got cells here, spongy mesophyll cells. There we are. I've drawn a few in. And we've got water vapour moving towards the stomatal pore, then you get this little cloud, as it were, little shell of water vapour, and we're getting, you know, diffusion away. So that's the diagram. Water molecules from the edge of the stomatal pores. So if I just highlight where the edge of this stomatal pore is, so there's the edge, and that would be in a circle. They evaporate more readily, so we getting more from the edge. Okay, and you can actually see, it doesn't really ask you for that, but you can see why this happens, because they've got they're nearer the air. They haven't got to diffuse through a load of other bits of water vapour, and so it's kind of like a steeper gradient there. And they've measured some of these things. They've measured the stomatal density. So we discussed this already. That's how many you get per millimetre squared. They've told us they're circles. Okay. And the total area of the pores was approximately the same. Okay. They've done one calculation for us, and we just need to repeat it to get the other answer. Calculate the total circumference of the stomatal pores for species B. Right, the first little trick here is, well, we're given an equation, so we're getting 2 pi r, but 2's fine, pi's fine, it's given us pi there, 3.14, times by r. What's r? So r is the radius, it tells us that, but have we got the radius anywhere? Oh, the nearest, but we've got, for any help, is a diameter. So if the diameter is 4, so diameter equals 4, then the radius must equal 2. So we are going to times that by 2. So that's our pi r, that's a 2 pi r. And then that'll be one of them. How many have we got? We've got 423. So we're going to times that by 423. So just again, that 2 pi r, that's the, if I draw another little diagram at the side here, that's telling us what the circumference, the edge of that circle is. And we've worked that out. And then we're going to, how many of those circles do we have in a square millimetre? So we're counting how many circles we've got, which is 423. And then you can work that out. So 2 times by 3.14 times by 423 equals 2656.44. Have I done that right now? So 2 times by 3.14 times by 2 times by 423. That's better. 5312. 5312.88. Now imagine doing all that work and not reading this last bit. Give your answer to 3 significant figures. A little maths trick on the end there. 3 significant figures. Yikes! What are we going to do? So we want to use these first 3. So 531. The one after it is 2. So 531, and we're just going to stick a 0 on there. 3 significant figures. That's horrible, isn't it? Anyway, 5310. Use these figures to identify which plant species A or B has the highest transpiration rate. I'm going to put this in the table here so we can compare it. Okay, so it told us somewhere which has the highest transpiration rate. It told us somewhere, here we go. Water molecules from the edges of stomatal pores evaporate more readily. So in that case it's the one with the most edge, one with the most circumference. So that's going to be B. And explain your answer. So you're not getting everything from B alone. You need to say, because it's got more circumference, more total circumference, got more total circumference, which is more edges of the stomata. And that's a lot of work for 14 marks.